Biodiversity genetic modification. Here's our objectives. First is the introduction of biodiversity genetic modification. Next is the history of genetic modification. And lastly, the usage and purpose of genetic modification. Here's the introduction of biodiversity genetic modification. First, what is biodiversity? Biodiversity refers to the range of living forms found in one region, such as animals, plants, fungi, and even microorganisms, such as bacteria that form our natural world. Each of these organisms and animals they interact with the ecosystem to preserve the balance and support life, much like an elaborated web. Everything in nature that we require to live that's supported by biodiversity, such as food, clean water, medicine, and shelter. Next, what is genetic modification? Genetic modification is a technique that involves transferring a pieces of DNA from one organism to another in order to change the characteristics of plant, animal, or microorganism. This is accomplished by selectively removing the desired genes from the DNA from one organism and inserting them into DNA of the other. This method has been used to create fungi and bacteria that produce, for example, biodiversity genetic modification, the history, usage, and process. In history of genetic modification, in 1973, Boyer and Cohen developed a genetic modification. Biochemist Herbert Boyer and Stanley Cohen developed genetic engineering by inserting the DNA of one bacteria into another. In 1974, Jenisch and Mint's first genetic modification animal, Jenisch created a transgenic mouse by introducing a foreign DNA into its embryo, and he studied the mammalian cells infected with Sumerian virus 40 rather, or what we call SB40. In 1982, FDA approves the first genetic modification organism pharmaceutical product, which they developed through genetic modification, a human insulin to treat diabetes. In 1994, transgenic tomatoes first released in the market. The first GMO produced created through genetic engineering, a genetic modification organism tomato becomes available for sale after studies evaluated by federal agencies that proved to be the safe as traditionally bred tomatoes. Usage of genetic modification. First is the medicine. Researchers tasked with the changing crops and microorganisms via genetic engineering, whether it's for agricultural or medi- medis- medicinal purposes, use extensive and careful screening and process to find specific molecule and proteins. Once selected, the desired genes are inserted into a host organisms and plants, eventually creating an ideal conditions for real-world usage. For potential drugs, those proteins and molecules must show promise in treating specific disease or condition. And for crops, they use those proteins and molecules must provide useful traits to plants without harming consumers and environment. Next, the usage of genetic modification in agriculture. Genetic modification provide a quicker and more precise ways to achieve the same goal in generation rather than 20. Genetically modified crops offer improved yields, enhanced nutritional value, longer shelf life, and resistant to drought, frost, or insect pests. Examples of genetic modification crops include corn varieties containing with a gene for bacterial pesticides that kills larval pests and soybeans with an inserted gene that renders them resistant to weed killers. Nutritionally enhanced GM crops under the development include varieties of wheat free of gluten and major cause of food allergy, vegetable with higher vitamin E content to help fight heart disease and golden rice. Genetically engineered to contain vitamin A and iron so as prevent common nutritional deficiency in developing countries. In the United States, genetical modification corn is used in many common foods including cornmeal, tortilla chips, and high fructose corn syrup, a sweetener in soft drinks and baked goods. Usage of genetic modification in animals and organisms. Genetic modification of an animal involves altering its genetic material by adding and changing of or removing a certain DNA sequences in a way of does not occur naturally. It aims a modify specific characteristics of an animal or introduce a new trait such as a disease resistance or enhanced growth. DNA is the genetic material of a organism and carries the instruction for all characteristics that an organism inherits. Changes introduced in an animal genetic makeup can therefore be transmitted to the next generation. While this technology has so far been used in plants for agriculture in microorganisms to produce enzymes and potential applications of genetic modification techniques to animals is also being researched. And here's the figure of producing a GM corp. First is the overview of the process of a bacteria into a DNA isolation into cloning genes and to designing genes, and it's transformed to a tissue culture and into a lastly into a plant breeding. So here are the process of genetic modification. The information about the microbe and its reproduction mechanisms are contained in its gene structure. The first step in the process of the identification of the desired trait from another organism, a gene containing this trait is first isolated and replicated. Next, the insertion of trait happens. What is transferred from one organism to another is not the whole gene, but only the section of gene that carry the particularly characteristics that will be integrated into a adult organisms. After that successful insertion, the modified organisms should be able to grow and replicate. In the process of recombinant DNA technology, the needed information of an organism can be borrowed from another, such as process means that farmer can design an organism to have characteristics necessary to address particular issues. However, because of the genetic modification organisms, biotechnology companies were also able to obtain the patient patent are novel life forms, which are the restrict their use as a result of companies that make GMOs could have the power of sue the farmers those fields that contaminated with these organisms even if is the result is inevitable, drifting from the neighboring fields by Nolson Nicholson, Nicholson 2014. The promise of better food that is more resistant to spoilage, pest invasiveness, and harsh weather conditions that made transgenic crops enticing to many people is truly debatable topic for the people of a 21st century given the promise of this technology and yet the fact is this very new. So I have a question for you. Genetic modification or rather engineering, it should be embraced as saving grace or it is to be feared as a possible threat to a sustainable food supply. So what do you think? And thank you for listening and I hope you learned something new about the genetic modification. And once again, thank you for listening and bye bye. Thank you.